This video is going to cover the entire immunology chapter starting on page 192. A few helpful tips while watching these videos is that if you would like to pause the video, hit the spacebar on your keyboard. To fast forward the video by 3 seconds, hit the right arrow key on your keyboard. And to go back 3 seconds in the video, hit the left arrow key. Immunology, lymphoid structures. What is a lymph node? A lymph node is an oval-shaped organ of the immune system which is distributed widely throughout the body. It has many afferent and one or more efferent lymphatic. Lymph nodes are also encapsulated with trabeculae. What is the function of afferent lymphatic in lymph nodes? Afferent lymphatics is what brings the material into the lymph nodes. What is function of efferent lymphatic? The function of efferent lymphatic is to take the material away from the lymph node. So remember, afferent brings the material in and efferent takes it out. And in a lymph node there are many afferent lymphatic and one or more efferent lymphatic. What are all the functions of lymph nodes? The functions of lymph nodes include non-specific filtration of macrophages, storage and activation of B and T cells, and antibody production. Briefly, describe the anatomy of the lymph node. To describe the anatomy of the lymph node, let's briefly take a look at the picture. We're going to start off talking about the capsule. As we mentioned earlier, that lymph node is an encapsulated organ, so therefore you see the capsule. Right underneath that is the subcapsular sinus. You also see many of the afferent lymphatics and one or more of the efferent lymphatic. You also see the medullary sinus, which contains the macrophages. There is the trabecula, along with medullary cords in which there are plasma cells. There is also the paracortex, which contains the T cell, and the follicle of the cortex, which contains the B cells. And lastly, each lymph node will have an artery and a vein. So to remember the layers from superficial to deep, remember there's the capsule first, followed by the subcapsular sinus, followed by the follicle of cortex, then the paracortex, and then the medullary cords and the medullary sinus. Lymph node follicle. What is lymph node follicle? Lymph node follicle is an area of the lymph node that is located in the outer cortex. It's the site of B cell localization and proliferation. What are the two types of follicles? The two types of follicles include primary follicles and secondary follicles. Describe the primary and secondary follicles. Primary follicles are dense and are dormant, so there's not a whole lot of activity that goes on in the primary follicles. But the secondary follicles have a pale germinal centers and are active. What is the innermost part of the lymph node? The innermost part of the lymph node is the medulla. What does the medulla consist of? The medulla consists of the medullary cords and the medullary sinuses. What are medullary cords? Medullary cords are closely packed lymphocytes and plasma cells. What cells are contained in the medullary sinuses? In the medullary sinuses, there are reticular cells and macrophages. What is the function of the medullary sinuses? The function of the medullary sinuses is to communicate with the efferent lymphatics. Where is the paracortex of the lymph node located? The paracortex of the lymph node is located between the follicles and the medulla. What does the paracortex contain? The paracortex contains the T cells. One thing to note about the paracortex is that it contains high endothelial venules through which B and T cells enter from bloodstream. So it's this region where the B cells and the T cells will enter into the lymph nodes. In what situation would the paracortex become enlarged? Paracortex becomes enlarged in extreme cellular immune response, such as with viral infections. What is the name of the condition in which the paracortex of the lymph node is not well developed? It's DeGeorge syndrome, in which the paracortex is not well developed. Lymph drainage. What is the primary lymph node drainage site for the upper limbs and the lateral breast? Upper limbs and lateral breast drains into the axillary lymph nodes. What is the primary lymph node drainage site for the stomach? The stomach drains into the celiac lymph nodes. What is the primary lymph node drainage site for the duodenum and the jejunum? Duodenum and jejunum drains into the superior mesenteric lymph nodes. What is the primary lymph node drainage site for the sigmoid colon? Sigmoid colon drains into the colic lymph nodes, which leads to inferior mesenteric lymph nodes. What is the primary lymph node drainage site for the lower portion of the rectum and the anal canal above the pectinate line? Lower portion of the rectum and the anal canal above the pectinate line drains into the internal iliac lymph nodes. What is the primary lymph node drainage site for the anal canal below the pectinate line? Anal canal below the pectinate line drains into the superficial inguinal lymph nodes. What is the primary lymph node drainage site for the testes? The testes drains into the superficial and the deep plexus, which then leads to the para-aortic lymph nodes. What is the primary lymph node drainage site for the scrotum? The scrotum drains into the superficial inguinal lymph nodes. What is the primary lymph node drainage site for the superficial part of the thigh? The superficial part of the thigh drains into the superficial inguinal lymph nodes. What is the primary lymph node drainage site for the lateral side of dorsum of foot? Lateral side of dorsum of foot drains into the popliteal lymph nodes. What all does the right lymphatic duct drain? The right lymphatic duct drains the right arm, right chest, and right half of the head. What all does the thoracic duct drain? The thoracic duct will drain everything else that is not covered by the right lymphatic duct. Briefly, describe spleen's anatomy. When talking about the anatomy of spleen, we will start off talking about the capsule. So here we see the capsule, the trabecula, we also see the vascular sinusoid, which is in the red pulp. Then we also see the white pulp, which consists of the follicle, marginal zone, and the periarterial lymphatic sheath, also known as PALS. A few additional notes is that the follicles contain the germinal center, in which there are B cells. Red pulp contains the red blood cells. Periarterial lymphatic sheath contains T cells. And marginal zone has the antigen presenting cells. Now let's zoom in further and talk about the sinusoids of the spleen. What are the sinusoids of spleen? Sinusoids of spleen are long vascular channels in the red pulp with fenestrated barrel hoop basement membrane, and macrophages are located near this area. Where are the T cells found in the spleen? The T cells are found in the periarterial lymphatic sheath, also known as PALS, within the white pulp of the spleen. Where are the B cells found? The B cells are found in the follicles within the white pulp of the spleen. What is one of the functions of macrophages of the spleen? One of the functions of spleen's macrophages is to remove encapsulated bacteria. Describe how splenic dysfunction leads to increase in susceptibility to encapsulated organisms. When you have splenic dysfunction, this leads to a decrease in immunoglobulin M. This will then lead to a decrease in the complement activation, which will lead to decrease in C3b opsonization, and therefore leading to that increase in susceptibility to encapsulated organisms. Name all the encapsulated organisms. Encapsulated organisms include Streptococcus pneumoniae, Haemophilus influenza type B, Neisseria meningitidis, Salmonella, Klebsiella pneumoniae, and lastly, Group B Streptococci. What is the mnemonic to help you remember all these encapsulated organisms? The mnemonic is to remember Shin sucks, and that's spelled as SHINSKS. So the first S is for Streptococcus pneumoniae, the HI is for Haemophilus influenza, N is for Neisseria meningitidis, the next S is for Salmonella, the K is for Klebsiella pneumoniae, and the last S is for Group B, Streptococci. So what can occur in patients post-splenectomy? In patients post-splenectomy, they lose their ability to ward off encapsulated organisms. They will also have Howell-Jolly bodies, which are nuclear remnants seen on the red blood cells. They will also form red blood cells into target cells, and thrombocytosis can also occur. What is a thymus? Thymus is an encapsulated immune organ which serves as a site of T cell differentiation and maturation. As a side note here, remember that T cells mature in the thymus, whereas B cells mature in the bone marrow. So remember T for T cells and T for thymus, and B for B cells and B for bone marrow. What is the thymus derived from? Thymus is derived from the third branchial pouches. A few additional notes about the anatomy of thymus is that the thymus consists of a cortex and medulla. The cortex is dense with immature T cells, whereas the medulla is pale and it contains mature T cells and epithelial reticular cells containing Hassel's corpuscles. Where does the positive and negative selection of T cells occur? The negative and positive selection of T cells occurs in the thymus's corticomedullary junction. Briefly, describe the positive and negative T cell selection. The positive and negative selection of T cells is what the thymus cells have to do. So basically, the thymocytes have to decide if the T cells are functional, which would be positive selection, or they need to be eliminated in case of autoreactive T cells, and this would be considered negative selection. We will talk further about this at a later time, but that's basically what positive and negative selection is. What is innate immunity? Innate immunity is also known as a non-specific immune reaction or the first-line defense. In innate immunity, there are germline encoder receptors that recognizes pathogens. The response that is seen in innate immunity is fast and it's non-specific. There's also no memory in innate immunity, so that means that anytime it encounters a pathogen, it reacts in a way if it's the first time it's encountering it. What are all the components of innate immunity? The components of innate immunity include neutrophils, macrophages, dendritic cells, natural killer cells of lymphoid origin, and complement. What is adaptive immunity? Adaptive immunity, also known as acquired immunity, is when there is memory that forms towards pathogens. It's an immune response that is slow on first exposure, but because memory develops, the memory response is faster and more robust. One additional thing to note about adaptive immunity is that when lymphocytes are developing, when it recognizes a pathogen, it will go through VDJ recombination. And when lymphocytes go through this VDJ recombination in response to a specific pathogen, it will have a more specific response in order to remove that pathogen. What are all the components of adaptive or acquired immunity? The components of adaptive or acquired immunity includes T cells, B cells, and circulating antibodies. Major histocompatibility complex 1 and 2. What is major histocompatibility complex? Major histocompatibility complex, or MHC, is a set of cell surface molecules that are encoded by the human leukocyte antigen gene, or the HLA gene. MHC's major function is to present processed antigen fragments to T cells and bind T cell receptors. What is MHC1 encoded by? MHC1 is encoded by HLA-A, HLA-B, and HLA-C. On what cells is MHC1 expressed on? MHC1 is expressed on all nucleated cells. That also means that MHC1 is not expressed on the red blood cells because red blood cells do not have a nucleus. What immunity is mediated by MHC1? MHC1 mediates viral immunity. What does MHC1 bind? MHC1 binds CD8 T cells on the T cell receptors. Describe how MHC1 initiates immune response. Since MHC1 mediates viral immunity, first you will have a cell that is infected by a virus. Now the antigens in that infected cell will combine with MHC1, and that will form the MHC1 antigen complex. This MHC1 antigen complex will then get expressed on the surface of the cell. Expression of this complex on the cell surface will then get recognized by T cell receptors and CD8 T cells, leading to the initiation of immune response. What is MHC2 encoded by? MHC2 is encoded by HLA-DR, HLA-DP, and HLA-DQ. What cells is MHC2 expressed on? MHC2 is expressed only on antigen-presenting cells. What type of immunity is mediated by MHC2? MHC2 mediates immunity to bacterial infections. What does MHC2 bind? MHC2 binds T cell receptors and CD4 T cells. Describe how MHC2 initiates an immune response. Since MHC2 mediates immunity to bacterial infection, first what happens is that the antigen-presenting cells will recognize the microbe, leading to the antigen fragments within the antigen-presenting cells to combine with MHC2. This will form the MHC2 antigen complex, which will then get expressed on the cell surface of these antigen-presenting cells. When this MHC2 antigen complex is expressed on these antigen-presenting cells, Here, the T cell receptors and the CD4 T cells will bind, and this leads to immune response. Human leukocyte antigen subtypes associated with diseases. What are the HLA genes? The HLA are gene family that provide instruction for making a group of related proteins known as the human leukocyte antigen or HLA complex. These HLA complexes then help the immune system distinguish between body's own proteins from proteins made by a foreign invader such as a virus or bacteria. What disease is the subtype HLA-A3 associated with? HLA-A3 is associated with hemochromatosis. Name all the diseases that the subtype HLA-B27 is associated with. HLA-B27 is associated with psoriasis, ankylosing spondylitis, inflammatory bowel disease, and Reiter syndrome. What is the mnemonic to help you remember all the diseases associated with HLA-B27 subtype? The mnemonic is just to remember the word pair. That's P for psoriasis, A for ankylosing spondylitis, I for inflammatory bowel disease, and R for Reiter syndrome. Name the disease associated with HLA subtype DQ2 or DQ8. The disease associated with HLA DQ2 or DQ8 is celiac disease. Name the diseases associated with the subtype HLA-DR2. The diseases associated with HLA-DR2 includes multiple sclerosis, hay fever, lupus, and Goodpasture syndrome. Name all the diseases associated with subtype HLA-DR3. The diseases associated with the subtype HLA-DR3 includes diabetes mellitus type 1 and Graves' disease. Name all the diseases associated with the subtype HLA-DR4. The diseases associated with HLA-DR4 includes rheumatoid arthritis and diabetes mellitus type 1. Name the diseases associated with the subtype HLA-DR5. HLA-DR5 is associated with Hashimoto's thyroiditis and pernicious anemia leading to vitamin B12 deficiency. What are the natural killer cells? Natural killer cells are a type of cytotoxic lymphocytes critical to the innate immune system. And remember, these are the only lymphocytes that are part of the innate immune system. Describe the action of the natural killer cells. Natural killer cells use perforin and granzymes to induce apoptosis, and in this way they kill off virally infected cells and tumor cells. Name all the cytokines responsible for activating natural killer cells or enhancing the activity of natural killer cells. The cytokines useful in this includes interleukin-2, interleukin-12, interferon beta, and interferon alpha. How does natural killer cells get activated to kill off cells? The way natural killer cells get activated to kill is when they are exposed to a non-specific activating signal on the target cells, or if there is an absence of class 1 MHC on the target cells. This will cause the activation of natural killer cells, which will eventually lead to apoptosis of virally infected cells and tumor cells. So basically, if there is an activating signal or if MHC class 1 is missing, this will cause the activation of natural killer cells, which would release its cytotoxic granules such as perforin and granzymes, and also induce the cytokine production to kill off its target cell. Major functions of B and T cells. What are the major functions of B cells? The major functions of B cells is to make antibodies, to mediate allergic reaction, cytotoxic and immune complex hypersensitivity, and it functions in case of hyperacute and humoral mediated acute and chronic organ rejection. Why does B cells make antibody? B cells make antibody to opsonize bacteria, neutralize virus through IgG antibody, activate complement through IgM and IgG antibody, and sensitize mast cells through IgE antibody. Which hypersensitivity reaction is allergy? Allergy is type 1 hypersensitivity via IgE antibody. What type of hypersensitivity is cytotoxic hypersensitivity? Cytotoxic hypersensitivity is type 2 hypersensitivity, and it's mediated via IgG antibody. What type of hypersensitivity is immune complex hypersensitivity? Immune complex hypersensitivity is type 3 hypersensitivity, which is also mediated via IgG antibody. Now let's move on to T cell functions. What is the function of CD4 T cells? The function of CD4 T cells is to help B cells make antibody and to produce cytokines to activate other cells of the immune system. What is the function of CD8 T cells? The function of CD8 T cells is to kill virus-infected cells directly. What type of hypersensitivity is the delayed cell-mediated hypersensitivity? The delayed cell-mediated hypersensitivity is type 4 hypersensitivity. And one last thing to note about T cells is that they function in acute and chronic cellular organ rejection. Differentiation of T cells. Describe the picture of T cell differentiation from the book. In this picture, first you see the T cell precursor. As we know, T cell starts off in the bone marrow, but it matures in the thymus. So from the bone marrow, it then goes to the thymus. Now once the T cell is in the thymic cortex, it has to go through positive selection. And positive selection is basically a check to see if the T cells are able to interact with MHC. And if they are able to interact with MHC, then it will receive surviving signals and therefore will survive. And if it's not able to interact with MHC, then it will receive no survival signal and therefore it will eventually die. And if it is able to interact with MHC, it depends on what MHC is able to interact with. If it's able to interact with MHC1, the cell becomes CD8 T cell or cytotoxic T cell. If it's able to interact with MHC2, then it becomes CD4 T cell, also known as helper T cell. So basically that summarizes the positive selection. Now let's move on to negative selection, which occurs in the medulla. So after the T cells have differentiated into CD4 T cells or CD8 T cells, it will now then go to the medulla to undergo negative selection. Here the negative selection will check how capable these new T cells are able to bind with self-MHC peptide. Here, if the interaction of the T cells is too strong with the MHC peptide, what will end up happening is the cell will now be told to undergo apoptosis. So this step is just to check if the interaction is just right and it's not too strong, because if it's too strong, what ends up happening is there is formation of self-reactive T cells that are capable of inducing autoimmune diseases. So that's why you want the interaction to be just right, and if it's too strong, then the cells will be told to undergo apoptosis. And once the T cells are able to pass both these checks of positive and negative selection, it will be released from the thymus into the lymph node, and it would now be a mature T cell. And if they are the mature CD8 T cells, they would now be called cytotoxic T cells, and if they are the mature CD4 T cells, they would now be called helper T cells. What are the functions of cytotoxic T cells? The function of cytotoxic T cell is to remove virus-infected cells, neoplastic cells, as well as donor graft cells. Describe the formation of T helper 1 and T helper 2 cells. If the helper T cell has the influence of interleukin 12, this cell will now become T helper 1 cell. If the helper T cell has the influence of interleukin 4, then the cell becomes T helper 2 cell. So interleukin 12 gives rise to T helper 1 cell, and interleukin 4 gives rise to T helper 2 cell. T and B cell activation. Name all the antigen-presenting cells. The antigen-presenting cells includes dendritic cells, macrophages, and B cells. Remember, dendritic cells are the only antigen-presenting cells that can activate naive T cells. Now, when talking about T cells and B cell activation, remember there are two signals that are required for T cell activation. Two signals are also required for B cell activation, and class switching also requires two signals. Describe the process of naive T cell activation. Please refer to the picture on your screens to understand naive T cell activation. On the left, you could see cytotoxic T cell activation, whereas on the right, you see the helper T cell activation. First, in both cases, you could see a foreign body being phagocytosed by dendritic cell or antigen-presenting cell. That foreign body antigen is now going to combine with MHC, and this will form the antigen-MHC complex. In case of cytotoxic T cell activation, you see the complex being formed between MHC1 plus the foreign antigen, whereas in the case of helper T cell activation, you see the complex being formed between MHC2 and the foreign antigen. Once the MHC and antigen complex has been formed, it will be now expressed on the cell surface. The MHC1 and antigen complex is going to be recognized by the T cell receptors on the cytotoxic T cell, whereas the MHC2 antigen complex is going to be recognized by the T cell receptors on the T helper cells. And this is the first signal. Also, in the picture here, you could see the co-stimulatory signal. And this co-stimulatory signal is given by the interaction of B7 and CD28, which makes up the second signal. When both of these signals are achieved, it will lead to activation of both T helper cells and cytotoxic T cells. The T helper cell is now going to be able to produce cytokines, whereas the cytotoxic T cell is able to recognize and kill virus-infected cells. Describe the process of B cell activation and class switching. Please refer to the picture on your screen to understand B cell activation and class switching. First, we have already talked about how T cells are activated, so that's already done with. Now let's talk about B cell activation and how that happens. Looking at top right of the picture, you could see that there is an antigen which is recognized by B cell receptor. Now there is going to be B cell receptor mediated endocytosis, so that antigen is now going to come inside the B cell. Now inside the B cell there is going to be complex formation between M8C2 and the foreign antigen. Once this complex has been formed, it's going to get expressed on the B cell surface, and that expression you can see here on the picture where it says M8C2 plus the peptide. This M8C2 and the antigen complex is now going to get recognized by the T cell receptors on the T helper cell, and this makes up the first signal. Now also on the picture here you could see that there is CD40 receptors on the B cell, which binds to the CD40 ligand, which is on the T helper cells. And this is your signal 2. Now when both of these signals are achieved, the T helper cells will now release its cytokines, and this will determine the Ig class switching of the B cell. So as you can see in the picture, all the interleukins are being released and it's being recognized by the interleukin receptor, which will cause B cell activation and make it undergo class switching, and this finally causes the B cells to produce appropriate antibodies. Helper T cells. As we know from earlier discussions that activated CD4 T cell will become T helper 1 and T helper 2 cells. What does T helper 1 cell secrete? T helper 1 cell secretes interferon gamma. What is the function of T helper 1 cell? The function of T helper 1 cell is to activate macrophages. What inhibits T helper 1 cells? T helper 1 cells are inhibited by interleukins that are released from T helper 2 cells. So just remember that both of these T helper cells are inhibiting one another by its cytokines. So T helper 1 is inhibited by interleukin 4 and interleukin 10 that is secreted by T helper 2 cells. What all does T helper 2 cells secrete? T helper 2 cells secrete interleukin 4, interleukin 5, interleukin 10, and interleukin 13. And remember, from those, interleukin 4 and interleukin 10 is what inhibits interleukin 1. What are the functions of T helper 2 cells? T helper 2 cells recruit eosinophils for parasite defense, and they also help out B cells because they help promote immunoglobulin E production by the B cells. What inhibits T helper 2 cells? As we mentioned earlier, both the T helper cells inhibit one another, so T helper 2 cell is inhibited by the interferon gamma that is secreted by T helper 1 cell. What is macrophage lymphocyte interaction? As we mentioned earlier that T helper 1 cell activates macrophages, also remember that macrophages can also activate T helper 1 cell. So this is basically what macrophage lymphocyte interaction is, that activated lymphocytes and macrophages stimulate one another. Cytotoxic T cells. What is the function of cytotoxic T cells? The function of cytotoxic T cell is to kill virus-infected, neoplastic, and donor graft cells by inducing apoptosis. Describe how cytotoxic T cells kills off all these different types of cells. Cytotoxic T cells will release cytotoxic granules containing performed proteins, such as porphyrin, which will help deliver the content of granules into the target cells. It also releases granzymes, which is a serine protease, and that activates apoptosis inside the target cell. And lastly, it also releases granulysin, which is an antimicrobial, and that also induces apoptosis. So basically, the way cytotoxic T cells kills off virus-infected cells, neoplastic cells, is basically by releasing these granules, and this will end up destroying the cell by inducing apoptosis. One additional thing to remember about cytotoxic T cell, which is something we've already mentioned several times, is that cytotoxic T cells have CD8, which binds to M8C1 on virus-infected cells. What are regulatory T cells? Regulatory T cells, which were formerly known as suppressor T cells, are a subpopulation of T cells which modulate the immune system. They maintain immune tolerance and prevent autoimmune diseases. It does all of this by producing anti-inflammatory cytokines like interleukin 10 and TGF-beta. So basically, as their name suggests, they regulate the immune system and make sure that it does not go crazy. One additional thing to note about regulatory T cells is that they express CD3, CD4, and CD25 as cell surface marker. You can also think of these cells as cells that will shut down the immune system after they have eliminated a pathogen, so this will prevent autoimmunity. Antibody structure and function. Briefly, describe the antibody structure. When looking at the picture of the antibody, remember the letter V stands for variable, and this is the amino terminal. The letter C stands for constant, and this is the carboxy terminal. H stands for heavy chain, and L stands for light chain. So for example, VH would be variable heavy chain, VL would be variable light chain, CL would be constant light chain, and CH would be constant heavy chain. So one antibody will have two of these, one on each arm. The heavy chain contributes to both Fc and the Fab fraction, whereas the light chains only contribute to Fab fraction. What part of the antibody does an antigen bind to? Antigen binds to the variable part of light and heavy chain. This is located on the Fab fraction, and there is only one antigenic specificity expressed per B cell. Where on the antibody does complement bind? Complement binding can occur on the Fc portion of the antibody, but it only happens for antibody IgG and IgM. Describe all the features of the Fc portion. As we mentioned already, Fc portion is the constant portion. They have a carboxy terminal. There is complement binding that also occurs here in some isotopes such as IgG and IgM. They have carbohydrate side chains, and this is the area that determines the isotype. So this is basically the area that decides what immunoglobulin it's going to be. What are all the functions of antibody? The function of antibody is opsonization, neutralization, and complement activation. Describe the opsonization process of antibody. When a pathogen enters the system, it gets covered with antibodies, and this will cause for it to get phagocytosed and removed from the body. Describe the neutralization process of antibodies. When a pathogen enters the body, the antibodies will surround it and neutralize it. And when this happens, the pathogen is no longer able to carry out its function. Describe the complement activation by antibodies. When antibodies recognizes a pathogen, it will activate the complement cascade. And with the complement cascade activated, it will enhance the opsonization and the lysis of the pathogen. So these are all the ways that antibodies fight off pathogens. Now one concept to remember here is that any pathogen that enters the system will cause an antibody response. So since there could be millions and billions of different pathogens, you need diversity in your antibodies to ward off all these different pathogens. So therefore, there are several different ways of antibody diversity because it has been estimated that humans generate about 10 billion different antibodies. So, name the 4 different ways of generating antibody diversity. The first way to achieve antibody diversity is through random recombination of VJ, which are the light chains, and VDJ, which are the heavy chain genes. The second way is for random recombination of heavy chains with light chains. The third way is somatic hypermutation, and the final way is the addition of nucleotides to DNA during recombination, which is achieved by the enzyme terminal deoxynucleotidyl transferase. Immunoglobulin isotypes. What immunoglobulin isotypes are expressed on the mature B lymphocytes? Mature B lymphocytes expresses IgM and IgD on their surface. Describe how the other immunoglobulin isotypes are formed. We already know from previous discussions that B lymphocytes will differentiate into plasma cells, and this happens due to gene rearrangement, and it's also mediated by cytokines and CD40 ligand. So when plasma cells is formed from B lymphocyte, this plasma cell will now be able to secrete IgA, IgE, and IgG. Name all the immunoglobulin isotypes. The immunoglobulin isotypes includes IgG, IgA, IgM, IgD, and IgE. What is the mnemonic to help you remember all these immunoglobulin isotypes? Just remember GAME-D, so that's G, A, M, E, and D. So GAME-D. What is the main antibody of the secondary or delayed response to an antigen? The main antibody for the delayed response to an antigen is immunoglobulin G. What is the main antibody for the primary immediate response to an antigen? The main antibody that produces the primary immediate response to an antigen is immunoglobulin M. Which is the most abundant immunoglobulin isotype? The most abundant immunoglobulin isotype is immunoglobulin G. What are all the functions of immunoglobulin G? The functions of immunoglobulin G is to fix complement. They can also cross the placenta, therefore providing infants with passive immunity. And their function also includes opsonizing bacteria and to neutralize bacterial toxins and viruses. Does immunoglobulin A fix complements? No, immunoglobulin A does not fix complement because as we mentioned earlier that only immunoglobulin G and immunoglobulin M are the only two antibodies that fixes complements. What are all the functions of immunoglobulin A? The functions of immunoglobulin A is to prevent attachment of bacteria and viruses to the mucous membrane. Immunoglobulin A can also cross the epithelium by transcytosis. This is also found in secretions such as in the tears, saliva, and mucus, and they are also in the early breast milk known as colostrum. Couple of additional notes about IgA is that it's a monomer in the circulation and a dimer when secreted. And secondly, it can also pick up secretory components from epithelial cells before secretion. What are all the functions of IgM? As we already mentioned earlier that the function of IgM is to produce primary immediate response to antigen. It also has the ability to fix complements, but it does not cross the placenta. We also said that IgM and IgD are the two immunoglobulins that are expressed on the mature B lymphocyte cell surface. A few additional notes about IgM is that it's a monomer on B cell or a pentamer. The shapes of the pentamers are also important because the shape of the pentamer is what allows the efficient trapping of free antigens out of the tissue while humoral response evolves. What is the function of immunoglobulin D? The function of immunoglobulin D is still unclear, but it's found on the cell surface as we have already mentioned along with IgM. So basically IgD and IgM are the two immunoglobulins that are found on the cell surface of many B cells and in the serum. What is the function of immunoglobulin D? As we talked about in a different section, that immunoglobulin D is what mediates allergic reaction. So it makes sense that immunoglobulin D will bind mast cells and basophils. They also cross-link when exposed to allergens, and it mediates immediate type 1 hypersensitivity through release of inflammatory modulators such as histamine. So if you recall that any allergic reaction will have an increased release of histamine, and that is typically what leads to signs of allergies such as red nose and flushing. So again, with the activation of IgE, this will lead to a hypersensitivity reaction that will cause the release of inflammatory mediators such as histamine. A few additional notes to mention about immunoglobulin E is that it mediates immunity to worms by activating eosinophils, and it has the lowest concentration in the serum. Antigen type and memory. Describe the thymus-independent antigens. The thymus-independent antigens are the antigens that are lacking a peptide component. When an antigen lacks the peptide component, it cannot be presented by the M8C to the T cells. And this occurs in the case of lipopolysaccharide from cell envelope of gram-negative bacteria and the polysaccharide capsular antigens. So when that peptide is not able to be presented to the T helper cells, this will still result in the release of antibodies, but there is no formation of immunologic memory. Describe the thymus-dependent antigens. The thymus-dependent antigens are antigens that contain a protein component, such as in case of diphtheria vaccine. So when the antigen does contain this protein component, there is class switching that occurs and an immunologic memory will form as a result of direct contact of B cell with a T helper cell CD40 and CD40 ligand interaction. So the memory is forming because of this interaction, and this is the signal 2 interaction that occurs between B cells and T helper cells. Immunology, immune response. Complement system. What is the complement system? The complement system is a system of interacting proteins that play a role in the innate immunity and inflammation. The goal of the complement system is to produce the membrane attack complex, and this is what is responsible for defending against gram-negative bacteria. What are the three different complement activation pathways? The three complement activation pathways include the classic pathway, alternative pathway, and the lectin pathway. How is the classic complement pathway activated? The classic complement pathway gets activated via immunoglobulin G and immunoglobulin M. As we mentioned earlier in our discussion that these are the only two immunoglobulins to which the complements can bind. What is the mnemonic to help you remember that the activation of the classic complement pathway is due to immunoglobulin G or immunoglobulin M? The mnemonic is just remember that GM makes classic cars, so that's immunoglobulin G and immunoglobulin M, which mediates the activation of the classic pathway. How does the alternative complement pathway get activated? The alternative complement pathway will get activated when the system recognizes microbial surface molecules. How does the lectin complement pathway get activated? The lectin complement pathway gets activated when the system recognizes mannose or other sugars on the microbial surface. Now before we move on to talk about functions of different complement proteins, let's quickly talk about all the things that go on in the different pathways to form that membrane attack complex. So please refer to the picture in your book as well as the picture on your screen to help you understand the different pathways. Now we will start off this discussion talking about the classic pathway. Now let's say you have a bacteria that enters the system. The antibodies in your system will recognize this bacterial antigens and form the antigen-antibody complex. Now as we know from earlier discussions that IgG and IgM antibodies have an area for complement binding. So to the antigen-antibody complex of IgG and IgM, the initial C1 complex will bind. This initial C1 complex also contains C1 proteases, which will help cleave C2 into C2b and C2a, and C4 into C4a and C4b. So from the production of these four complement proteins, two of them are going to be released as anaphylatoxin, which is C2b and C4a. And these anaphylatoxins are what would be responsible for anaphylaxis if there are great amounts of these anaphylatoxins. Now the remaining two complement protein, which is C2a and C4b, will combine together to form C4b, C2a, which is also known as C3 convertase. C3 convertase will then cleave C3 and form C3a and C3b. C3a gets released in the system as an anaphylatoxin, whereas C3b will combine again with that C3 convertase and form C5 convertase. So C5 convertase is the combination of C4b, C2a, and C3b. This C5 convertase will then cleave C5 and form C5a and C5b. The C5a gets released in the system as an anaphylatoxin, whereas the C5b will act to initiate formation of the membrane attack complex. So for this, C5b will combine with C6, C7, C8, and C9, and it will form the membrane attack complex on the cell membrane of the bacteria. So when this membrane attack complex is formed, it makes a hole in the bacterial cell membrane, therefore killing the bacteria. Now let's move on to the next pathway, which is the alternative pathway of complement system. Now as we mentioned earlier that the alternative pathway is activated when the system recognizes the microbial surface molecule. So this is the way the system will detect the bacteria and start the alternative pathway. Now in this pathway you're starting off with C3 protein. C3 is an abundant protein that is found in your plasma, and this C3 protein will spontaneously hydrolyze to form C3b and C3a. C3a gets released in the system as an anaphylatoxin, and as we mentioned earlier, an anaphylatoxin is what mediates anaphylaxis if the anaphylatoxins are in great number. Now at this point you are left with C3b, which has to combine with something else. So for this you have another plasma protein called factor B. Factor B is going to get cleaved by factor D. This will form factor BB, which is written in your books as factors big B, little b. And then this factor BB will combine with C3b that we had left over, and this will form C3 convertase. And this C3 convertase is a different C3 convertase than what we mentioned in the classic pathway, because this C3 convertase is made up of C3b and the factor BB. Now this C3 convertase will cleave more of that C3, which is abundant in your plasma, and that leads to the formation of more C3a, which again we mentioned is an anaphylatoxin, and more of C3b. Now this C3b that is now made will go back and combine with that C3 convertase, which will now give you C5 convertase, which is made up of C3b, factor Bb, and another C3b. This C5 convertase will then cleave C5 and form C5a and C5b. The C5a gets released in the system as an anaphylatoxin, whereas the C5b will act to initiate formation of the membrane attack complex. So for this, C5b will combine with C6, C7, C8, and C9, and it will form the membrane attack complex on the cell membrane of the bacteria. So when this membrane attack complex is formed, it makes a hole in the bacterial cell membrane, therefore killing the bacteria. Alright, so now since we're done with the discussion on the classic and the alternative pathway, let's move on to the last pathway, which is the lectin pathway. Now the lectin pathway is similar to the classic pathway, it just has a different starting process, but everything else is the same as the classic process. So let's just start talking about the starting process, and eventually we will understand how it links up to the classic pathway. So in the classic pathway we have the antigen-antibody complex that activates that system, but the lectin pathway only gets activated when it recognizes sugars on the microbial surface. So if a bacteria that enters the system has a sugar on its surface such as mannose, the system will recognize this and start the lectin pathway. The lectin pathway starts off with something called MBL, or mannan-binding lectin. This is a protein that you have readily available in your plasma, and when it recognizes a microbial surface having a sugar such as mannose, this will go out and bind to it. So at this point you have mannose, which is bound to mannan-binding lectin, and this will then combine with MASP1 and 2, which is mannose-binding lectin-associated serine protease. And when this happens, this gives you something called C1-like complex. So the collective combination of the mannose sugar with the mannan-binding lectin plus the MAPS1 and 2 will give you the C1-like complex, and this C1-like complex will perform the action similar to C1 protease that we saw in the classic pathway. Alright, so now since all the pathways are now done, let's talk about some of the functions of some of these proteins. What is the function of C3b? The function of C3b is for opsonization. What is the function of C3a and C5a? As we mentioned, C3a and C5a along with other proteins such as C2b and C4a are all for anaphylaxis because they are all anaphylatoxins. What is another function of C5a? Another function of C5a is for neutrophil chemotaxis. It's a major chemoattractor from the complement system as it signals for neutrophil chemotaxis. What is the function of C5b, C6, C7, C8, and C9? All these proteins make up the membrane attack complex, and collectively they will lead to cytolysis of the bacterial cell membrane. What are some mnemonics to help you remember some of these functions of the complement proteins? The mnemonic, just remember, C3a and C5a both have the letter A, and A stands for anaphylaxis. C3b is for opsonization, so remember C3b binds bacteria. What are the two primary opsonins of our body's bacterial defense? The two primary opsonins are C3b and IgG. One additional thing to remember about C3b is that it also helps clear immune complexes. What are the two inhibitors of the complement activation on self cells such as on red blood cells? The two inhibitors of complement activation on self cells includes decay accelerating factor, or DAF, as well as C1 esterase inhibitor. Complement disorders. What is C1 esterase inhibitor deficiency? As we know from earlier, C1 esterase inhibitor will help prevent complement activation on self cells, such as on red blood cells. So therefore, if you are deficient in the C1 esterase inhibitor, this means there will be activation of complement on self cells, which would lead to a condition called hereditary angioedema. What is hereditary angioedema? Hereditary angioedema is a rare autosomal dominant inherited blood disorder that causes episodic attacks of swelling that may affect the face, extremities, genitals, GI tract, and the upper airways. What is the name of the other complement disorder that leads to activation of complements against red blood cells? The other disorder is decay accelerating factor, or GPI anchored enzyme deficiency. Describe the consequences that occurs with decay accelerating factor deficiency. As we mentioned earlier, that decay accelerating factor along with C1 esterase inhibitor helps prevent complement activation on self cells. So therefore, decay accelerating factor deficiency would lead to complement-mediated lysis of the red blood cells and lead to paroxysmal nocturnal hemoglobinuria, or PNH. What is C3 deficiency? C3, as we mentioned earlier, is normally very abundant in the plasma. It's used in both alternative and classic pathway. So if you're deficient in C3, this would lead to a lot of problems downstream in the formation of the membrane attack complex. And because of all this, it will lead to severe recurrent pyogenic sinus and respiratory tract infection. Deficiency of C3 will also increase the susceptibility to type 3 hypersensitivity reaction. What would be the consequence in C5 to C9 deficiencies? As we know from earlier, C5 to C9 is needed to make up the membrane attack complex, so when this is not able to happen, it would lead to recurrent Neisseria bacteremia. Important cytokines. Name all the cytokines secreted by the macrophages. The cytokines secreted by macrophages is interleukin 1, interleukin 6, interleukin 8, interleukin 12, and TNF-alpha. What are the two cytokines that is secreted by all T cells? The two cytokines secreted by all T cells is interleukin 2 and interleukin 3. Name the cytokine that is secreted by T helper 1 cell. The cytokine secreted by T helper 1 cell is interferon gamma. Name all the cytokines secreted by T helper 2 cells. T helper 2 cells secrete interleukin 4, interleukin 5, and interleukin 10. So remember, macrophages and T cells are the two most important cells that secrete cytokines. What is interleukin 1? Interleukin 1 is an endogenous pyrogen that causes fever and mediates acute inflammation. It also has the function to activate endothelium to express adhesion molecules, as well as to induce chemokine secretion to recruit leukocytes. What is interleukin 6? Interleukin 6 is an endogenous pyrogen which is also secreted by T helper 2 cells. It causes fever and stimulates the production of acute phase proteins. What is interleukin 8? Interleukin 8 is a major chemotactic factor to recruit neutrophils. What is the mnemonic to help you remember the functions of interleukin 8? The mnemonic, just remember, clean up on aisle 8. That should help you remember that neutrophils are recruited by interleukin 8 to clear off infections. What are all the functions of interleukin 12? The functions of interleukin 12 is to induce differentiation of T cells into T helper 1 cell, as well as to activate natural killer cells. One additional thing to remember about interleukin 12 is that they are also secreted by B cells. What are all the functions of TNF-alpha? The functions of TNF-alpha is to mediate septic shock. They also activate endothelium and cause leukocyte recruitment. Now we will move on to the important cytokines that are secreted by T cells. What is the function of interleukin 2? The function of interleukin 2 is to stimulate the growth of helper, cytotoxic, and regulatory T cells. What is the function of interleukin 3? The function of interleukin 3 is to support the growth and differentiation of bone marrow stem cells. They have functions similar to that of granulocyte macrophage colony stimulating factor. Also remember that these two interleukins, interleukin 2 and 3, are secreted by all T cells. What is the function of interferon gamma? The function of interferon gamma is to activate macrophages and T helper 1 cell. Interferon gamma's function is also to suppress T helper 2 cells and has antiviral and anti-tumor properties. What is the function of interleukin 4? The function of interleukin 4 is to induce the differentiation of T helper 2 cells. They also promote the growth of B cells and enhance class switching to IgE and IgG. What is the function of interleukin 5? The function of interleukin 5 is to promote differentiation of B cells. They also enhance class switching to IgA and stimulate the growth and differentiation of eosinophils. What is the function of interleukin 10? The function of interleukin 10 is to modulate inflammatory response. Interleukin 10 can also inhibit the activation of T cells and T helper 1 cells. An additional fact to note about interleukin 10 is that they are also secreted by regulatory T cells. Name a protein that has an action similar to that of interleukin 10. A protein that has an action similar to that of interleukin 10 is TGF-beta, or transforming growth factor beta, and the reason for this is because it is involved in the inhibition of inflammation. What is the mnemonic to help you remember the functions of some of the interleukins we just talked about? The mnemonic is to remember hot T-bone steak, and that will help you remember the functions of interleukin 1 through interleukin 5. Hot is for fever, and that is one of the actions of interleukin 1. T is for T cells because interleukin 2 stimulates T cells. Bone is for bone marrow because interleukin 3 stimulates the bone marrow. The letter E in the word steak is for IgE because interleukin 4 stimulates IgE production. And A in the word steak is for IgA because interleukin 5 stimulates IgA production. Interferon mechanism. What are interferons? Interferons are proteins made and released by the host cell in response to the presence of pathogen. These interferons basically just take the uninfected host cells and puts them in an antiviral state. Interferons do this by inducing the production of ribonucleases that inhibit viral protein synthesis by degrading viral mRNA, but it does not affect the host mRNA. What is the mnemonic to help you remember the actions of interferon? Just remember, interferons interferes with viruses. Name the primary functions of all the interferons. The primary functions of alpha and beta interferon is to inhibit viral protein synthesis, whereas the primary function for gamma interferon is to increase the MHC1 and MHC2 expression and antigen presentation in all cells. Gamma interferons can also activate natural killer cells to kill virus-infected cells. What are cell surface proteins? As the name suggests, cell surface proteins are proteins that are on the surface of cells, and all cells except for red blood cells have MHC1 as their cell surface protein. And the reason MHC1 is not on the cell surface of red blood cells is because of the fact that red blood cells do not have a nucleus, since MHC1 is only expressed on all nucleated cells. What are all the cell surface proteins seen on T cells? The cell surface protein of T cells include T cell receptor, which binds the antigen MHC complex. They also have CD3, which is associated with T cell receptor for signal transduction. And lastly, they also have CD28, which binds to the B7 of antigen-presenting cells. What are the cell surface proteins seen on helper T cells? The cell surface proteins for helper T cells include CD4 and CD40 ligand. CD40 ligand is what binds to CD40 on B cells. What is the cell surface protein for cytotoxic T cells? As we know from earlier discussions that cytotoxic T cells have CD8. What are the cell surface proteins for B cells? Cell surface proteins for B cells includes immunoglobulin, which binds antigens. B cells also have CD19, CD20, and CD21. CD21 is the receptor for Epstein-Barr virus. Cell surface protein of B cells also includes CD40, which binds to the CD40 ligand of helper T cells. There's also MHC2 and B7. B7 is what binds to CD28 of T cells. What is the mnemonic to help you remember that CD21 is the receptor for Epstein-Barr virus? The mnemonic is to remember that you can drink beer at a bar when you're 21. So remember B cells, Epstein-Barr virus, CD21. Name all the cell surface proteins of macrophages. The cell surface proteins for macrophages include CD14, CD40, B7, MHC2 because macrophages are a type of antigen-presenting cells. Macrophages also have Fc and C3b receptors to enhance phagocytosis. What are all the surface proteins seen in natural killer cells? Natural killer cells have CD16, which binds the Fc portion of immunoglobulin G, and CD56, which is a unique marker for natural killer cells. What is anergy? Anergy is a term in immunobiology that describes a lack of reaction by the body's defense mechanism to foreign substances. So in this, a self-reactive T cell becomes non-reactive without a co-stimulatory molecule, and B cells can also become anergic, but tolerance is less complete than in T cells. Effects of bacterial toxins. This part is important to know because some bacterial toxins can sometimes cause inappropriate activity of the host immune system, which can lead to massive autoimmune reactions, and this can also lead to shock. Describe the immune reaction that may occur when exposed to superantigens such as Staph aureus and Streptococcus pyogenes. Exposure to superantigens such as Streptococcus pyogenes and Staph aureus can lead to cross-linkage of the beta region of T cell receptor to the MHC2 of antigen-presenting cells, and this can activate any T cells, leading to a massive release of various cytokines, and this can cause autoimmune reaction as well as shock. Describe the immunologic effects of endotoxins or lipopolysaccharides that are released by gram-negative bacteria. Endotoxins or lipopolysaccharides of gram-negative bacteria can directly stimulate macrophages by binding to endotoxin receptor CD14. As we mentioned earlier, macrophages have CD14 receptors, and this is what recognizes endotoxins, and this will cause a overstimulation of macrophages, which can cause a massive autoimmune reaction. So one thing to note here is that in this situation there is going to be a direct stimulation of macrophages, so T helper cells are not involved. Antigen variation. What is antigenic variation? Antigenic variation refers to the mechanism by which infectious organisms such as protozoas, bacteria, and viruses will alter their surface protein in order to evade a host immune response. So basically, this is the organism's way of protecting itself from being killed by the host's immune system. Describe some mechanisms of antigenic variation seen in pathogens. To avoid immune attacks from the host, a pathogen may go through DNA rearrangement and RNA segment reassortment. This is what is seen in influenza when it goes through its major shift. Give some examples of antigenic variation seen with some pathogens. Bacteria such as Salmonella can have variations in their flagella, and Neisseria gonorrhea can make changes in their pilus protein. Viruses such as influenza can go through a major shift or a minor drift. Parasites such as trypanosomes can go through programmed rearrangement. So these are all different examples of antigenic variations. Passive and active immunity. We have already talked about innate and adaptive immunity. Now let's talk a bit about passive and active immunity. What is passive immunity? Passive immunity, as the name suggests, it's immunity that is passed from one person to another. So basically, in passive immunity, one person is given antibodies of another person. Give some examples of passive immunity. Examples of passive immunity is when you get immunoglobulin A from breast milk, antitoxin injections, and humanized monoclonal antibody. What is the means of acquisition of passive immunity? As we already mentioned, the means of acquisition is to receive performed antibodies. Is the onset of passive immunity rapid or slow? The onset of passive immunity is rapid. What is the duration of passive immunity? The duration of passive immunity is for a short span. It has a half-life of about 3 weeks. Give some examples of situations in which you would want to give performed antibodies to create passive immunity. Some situations in which you would need to give performed antibodies would be after exposure to tetanus toxin, botulinum toxin, hepatitis B, and rabies virus. What is the mnemonic to help you remember some of these conditions in which you would want to give performed antibodies? The mnemonic, just remember, to be healed rapidly. So you would give performed antibodies to create passive immunity when you would want a person to be healed rapidly. So that's 2 for tetanus toxin, B is for botulinum toxin, healed is for hepatitis B virus, and rapidly is the rabies virus. What is active immunity? Active immunity is what happens when you're exposed to a foreign antigen for the first time. When this happens, B and T cells are activated and they form memory B and T cells. And these memory cells will then be able to remember each specific pathogen that was encountered, and they will mount a strong response if the pathogen is ever detected again. What is the means of acquisition of active immunity? Again, as we already mentioned, active immunity starts when you're exposed to a foreign antigen for the first time. Describe the onset of active immunity. Active immunity has a slow onset because it takes time for memory to develop against a certain antigen. Describe the duration of active immunity. Active immunity lasts a long time because there is memory that is formed against that antigen. What are some examples of active immunity? Examples of active immunity would include natural infections, vaccines, and toxoids. These are all different ways in getting active immunity, so if any time that pathogen is ever encountered again, the body will fight it without any interruptions because it has a memory towards that pathogen. One additional thing to note is that passive and active immunizations can be given in case of hepatitis B or rabies exposure. Vaccination. What is the use of vaccination? Vaccines are used to induce an active immune response, humoral and/or cellular, to a specific pathogen. So basically the reasons you're given vaccines is that anytime you encounter that pathogen again, your body already has memory towards it, and therefore it will be able to counter it really fast without causing any disruptions. What are the 2 different types of vaccines? The 2 different types of vaccines includes live attenuated vaccine and inactivated or killed vaccine. What is live attenuated vaccine? Live attenuated vaccine is when you create a vaccine in which the infectious agent is still alive, but it has lost its pathogenicity. So basically, live attenuated vaccine is when you're keeping the microorganism alive, but you have made it harmless. What type of immunity are you trying to induce when giving live attenuated vaccine? You're trying to induce cell-mediated immunity. What are the pros and cons of live attenuated vaccine? The pros of live attenuated vaccine is that it would induce a strong, often lifelong immunity towards that pathogen. The cons of live attenuated vaccine would be that that organism may become virulent again, so it may revert back to a harmful form. Give some examples of live attenuated vaccine. Live attenuated vaccines are what are used to protect against measles, mumps, polio, rubella, varicella, and yellow fever. What is inactivated or killed vaccine? Inactivated or killed vaccine is when you have a vaccine that is formed after you take a virus and you kill it by methods such as heat or formaldehyde. So you kill the virus to a point where it can no longer replicate, but the virus capsid proteins are still intact, and they are intact enough to be recognized by the immune system to evoke a response. What type of immunity are you trying to induce when giving inactivated or killed vaccine? Inactivated or killed vaccine will induce a humoral immunity, also known as antibody-mediated immunity. What are the pros and cons of inactivated or killed vaccine? The pros of inactivated or killed vaccine is that they are stable and safer than live vaccines, and the cons are that they are a weaker immune response, so booster shots are usually required. Give some examples of inactivated or killed vaccines. Examples of inactivated or killed vaccines include cholera, hepatitis A, polio, and rabies vaccines. Hypersensitivity reactions. What is type 1 hypersensitivity reaction? Type 1 hypersensitivity reaction is an allergic reaction provoked by re-exposure to a specific type of antigen, referred to as an allergen. This type of allergic reaction is considered to be anaphylactic and atopic. Define atopic or atopy. Atopy or atopic syndrome is when somebody has a predisposition towards developing a certain type of hypersensitivity reaction. To help understand this, just think about how people are allergic to peanuts and things like that. This would be a type 1 hypersensitivity reaction. Describe what all occurs to cause type 1 hypersensitivity reaction. To cause type 1 hypersensitivity reaction, free antigens cross-link with immunoglobulin E on pre-sensitized mast cells and basophils. This triggers the release of vasoactive amines such as bradykinin and histamine, and they act on postcapillary venules. Type 1 hypersensitivity reaction develops rapidly after antigen exposure because of the performed antibodies. What is the mnemonic to help you remember type 1 hypersensitivity reaction? The mnemonic is just to remember first and fast. So this is the first type of hypersensitivity reaction, and it's fast because it causes anaphylaxis. One additional side note here is that remember that type 1, type 2, and type 3 are all antibody mediated, whereas type 4 hypersensitivity reaction is cell mediated. What test would you need to do to diagnose type 1 hypersensitivity? To diagnose type 1 hypersensitivity, you do a skin test for a specific IgE. Give some examples of type 1 hypersensitivity reaction. Type 1 hypersensitivity reaction includes asthma, hives, drug allergies, and food allergies. What is type 2 hypersensitivity reaction? Type 2 hypersensitivity reaction is cytotoxic or antibody-mediated hypersensitivity reaction. In this, IgM or IgG will bind to fixed antigen on enemy cells, leading to the cellular destruction. What are the three different mechanisms of type 2 hypersensitivity? The first mechanism is opsonization, which will lead to phagocytosis and complement activation. In this, basically, the antibodies will tag the pathogen, and that pathogen will then get opsonized and phagocytosed. The second mechanism is the complement-mediated lysis, in which there is formation of the membrane attack complex, and that destroys the cell membrane, killing the pathogen. And the third mechanism is the antibody-dependent cell-mediated cytotoxicity, or ADCC, which is usually due to natural killer cells. In this, what ends up happening is that IgG or IgM antibody will tag the foreign antigen, which would then be killed by natural killer cells. Give an example of type 2 hypersensitivity reaction. An example of type 2 hypersensitivity reaction is incompatible blood transfusion. So basically what would happen in this is that the antibodies will react with the incompatible blood, leading to lysis of those cells. What test is used to diagnose type 2 hypersensitivity? The test useful in diagnosing type 2 hypersensitivity is direct and indirect Coombs test to detect the antibodies. What is type 3 hypersensitivity? Type 3 hypersensitivity is when antigen-antibody complexes are formed that are not adequately cleared by the innate immune cells, so therefore these antigen-antibody complexes will start accumulating, giving rise to an inflammatory response and attraction of leukocytes. So basically, type 3 hypersensitivity is when you have formation of immune complexes. What is the mnemonic to help you remember type 3 hypersensitivity? To help you remember immune complexes are what occurs in type 3 hypersensitivity, just remember that immune complexes are three things stuck together: antigen, antibody, and complement. What are the two type 3 hypersensitivity reaction? The two type 3 hypersensitivity reaction includes serum sickness and Arthus reaction. What is serum sickness? Serum sickness is a type of immune complex hypersensitivity or type 3 hypersensitivity reaction. In this condition, what ends up happening is that when an anti-serum is given, which might be derived from a non-human animal source, the human immune system might mistake the proteins present for harmful antigens, and therefore it will react to it by producing antibodies, which will combine with these proteins to form immune complexes. These immune complexes will then enter the wall of the blood vessels and initiate an inflammatory response, and therefore leading to that type 3 hypersensitivity. A few additional things to remember about serum sickness is that now most of the serum sickness is caused by drugs and not serum. Signs and symptoms of serum sickness includes fever, urticaria or hives, arthralgias, proteinuria, and lymphadenopathy 5 to 10 days after antigen exposure. What is Arthus reaction? Arthus reaction is a local subacute antibody-mediated hypersensitivity, which is a type 3 hypersensitivity reaction. In this, what ends up happening is that intradermal injection of antigens induce antibodies, which will then form the antigen-antibody complexes in the skin. This condition is characterized by edema, necrosis, and activation of complement. What test is used to diagnose type 3 hypersensitivity? The test used to diagnose type 3 hypersensitivity is immunofluorescence staining, because in this you are trying to examine a tissue biopsy for deposits of immunoglobulins and complement, and you do this by immunofluorescent microscopy. What is type 4 hypersensitivity? Type 4 hypersensitivity, which is also known as delayed T-cell mediated hypersensitivity, is the only hypersensitivity that is not antibody mediated, but rather a cell mediated reaction. In this hypersensitivity, what happens is that sensitized T lymphocytes will encounter an antigen, and then it will cause a release of lymphokines. This will lead to macrophage activation. What are some examples of type 4 hypersensitivity? Some examples of type 4 hypersensitivity includes transplant rejection as well as contact dermatitis. What is the mnemonic to help you remember that type 4 hypersensitivity is the delayed type hypersensitivity? The mnemonic is to remember fourth and last, and this is the delayed. So the last one is the delayed hypersensitivity reaction. What are the 4 T's to help you remember type 4 hypersensitivity? The 4 T's includes T lymphocytes, transplant rejection, TB skin test, and touching to help you remember contact dermatitis. What are the diagnostic tests for type 4 hypersensitivity? The diagnostic test includes PPD and the patch test. PPD is what they do for contact dermatitis, whereas the patch test is the TB skin test. What is the mnemonic to help you remember all 4 of these hypersensitivities? The mnemonic is to remember the word acid. A stands for anaphylactic and atopic for type 1 hypersensitivity. C stands for cytotoxic or antibody mediated, which is that type 2 hypersensitivity. I stands for immune complex or type 3 hypersensitivity. And D is for delayed or cell-mediated type 4 hypersensitivity. Hypersensitivity disorders. Name all the type 1 hypersensitivity reactions. The type 1 hypersensitivity reaction includes anaphylaxis due to bee stings and some food or drug allergies, such as peanut allergy, as well as allergic or atopic disorders such as rhinitis, hay fever, eczema, hives, and asthma. One thing to note about type 1 hypersensitivity reaction is that they happen immediately after exposure to the antigen. Name all the diseases that can occur due to type 2 hypersensitivity reaction. Type 2 hypersensitivity reaction can lead to autoimmune hemolytic anemia or AIHA. It can lead to pernicious anemia due to the immune system attacking the parietal cells. Also idiopathic thrombocytopenic purpura, erythroblastosis fetalis, acute hemolytic transfusion reaction, rheumatic fever, Goodpasture syndrome, bullous pemphigoid, and pemphigus vulgaris are all examples of conditions that can occur due to type 2 hypersensitivity reaction. One thing to note here about type 2 hypersensitivity reaction is that the disease tends to be specific to a tissue or site where the antigen is found. So the attack happens at a local area, and the disease also occurs at that same specific area. Name some diseases that can occur due to type 3 hypersensitivity reaction. Type 3 hypersensitivity reaction can lead to lupus, polyarthritis nodosa, post-streptococcal glomerulonephritis, serum sickness, and Arthus reaction. And a classic example of Arthus reaction is swelling and inflammation that is seen following a tetanus vaccine. One additional thing to remember about type 3 hypersensitivity reaction is that this reaction occurs due to formation of the immune complexes, as we mentioned earlier, and these immune complexes can enter the wall of the blood vessels. So that's why type 3 hypersensitivity reaction is associated with vasculitis and systemic manifestations. Give some examples of type 4 hypersensitivity reaction. Examples of type 4 hypersensitivity reaction include multiple sclerosis, Guillain-Barré syndrome, graft versus host disease, PPD test for tuberculosis, which requires a few days of waiting because it's a type 4 hypersensitivity reaction, which is the delayed response it's waiting for. So this requires a few days of waiting before the test can be read. And lastly, the type 4 hypersensitivity reaction also includes contact dermatitis due to poison ivy and nickel allergy. A few additional notes about type 4 hypersensitivity is first to remember that poison ivy is a type 4 hypersensitivity and not a type 1, because other plant-related conditions such as hay fever are type 1 hypersensitivity. So remember, poison ivy is in fact type 4 hypersensitivity. Additional facts about type 4 hypersensitivity is that this is a delayed response and it does not involve antibodies. But all the other hypersensitivity reactions such as type 1, type 2, and type 3 are all antibody-mediated. Blood transfusion reactions. Name all the different types of blood transfusion reaction. The different types of blood transfusion reaction that can occur includes an allergic reaction, anaphylactic reaction, febrile non-hemolytic transfusion reaction or FNHTR, or acute hemolytic transfusion reaction or HTR. Describe the allergic reaction seen in blood transfusion. The allergic reaction of blood transfusion is a type 1 hypersensitivity reaction in which there is a mild reaction to the plasma proteins of the transfused blood. Describe the clinical presentation seen with allergic reaction due to blood transfusion. The clinical presentation in this case includes urticaria or hives, pruritus or itching, wheezing, and fever. How can allergic reaction due to blood transfusion be treated? Mild allergic reactions can be treated with antihistamines. Describe the anaphylactic reaction seen with blood transfusion. Anaphylactic reaction is an allergic reaction that is more severe and it can also lead to shock. Anaphylactic reaction can occur if a person that is deficient of IgA is given blood products that contain IgA. So you should never do this, otherwise it will lead to anaphylactic reaction. So therefore, IgA deficient individuals must only receive blood products that lack IgA. Describe the clinical presentation seen in anaphylactic reaction. The clinical presentation includes dyspnea, bronchospasms, hypotension, respiratory arrest, and shock. What is febrile non-hemolytic transfusion reaction or FNHTR? FNHTR is a type 2 hypersensitivity reaction. In this, the host antibodies will attack the donor's HLA antigens and leukocytes. Describe the clinical presentation you see in febrile non-hemolytic transfusion reaction. The clinical presentations seen with FNHTR are the nonspecific symptoms such as fever, headaches, chills, and flushing. What is acute hemolytic transfusion reaction or HTR? HTR is a more severe form of FNHTR. This is also a type 2 hypersensitivity reaction. In this reaction, there can be intravascular hemolysis due to the ABO blood group incompatibility, or extravascular hemolysis due to host antibody reaction against foreign antigen on donor red blood cells. Describe the clinical presentation of acute hemolytic transfusion reaction or HTR. The clinical presentation of HTR includes fever, hypotension, tachypnea, tachycardia, flank pain, and hemoglobinuria, which is seen with intravascular hemolysis, or jaundice, which is seen in extravascular hemolysis. Antibodies and their associated disorder. There's not much to explain here, it's just a list of different antibodies and the disorders they're associated with. But it is a high-yield topic, so it's worth knowing. What disorder is anti-nuclear antibodies associated with? Anti-nuclear antibodies, or ANA, are associated with lupus. What disorder is anti-double-stranded DNA or anti-Smith antibodies associated with? Anti-double-stranded DNA or anti-Smith antibodies are also associated with lupus. What condition is anti-histone antibodies associated with? Anti-histone antibodies are associated with drug-induced lupus. What disorder is rheumatoid factor and anti-cyclic citrullinated peptide antibodies associated with? Rheumatoid factor and anti-CCP are associated with rheumatoid arthritis. What disorder is anti-centromere antibodies associated with? Anti-centromere antibodies are associated with scleroderma or CREST syndrome. What disorder is anti-SCL70 or anti-DNA topoisomerase I antibodies associated with? Anti-SCL70 or anti-DNA topoisomerase I antibodies is associated with diffuse scleroderma. What disorder is anti-mitochondrial antibodies associated with? Anti-mitochondrial antibodies associated with primary biliary cirrhosis. What disorder is IgA anti-endomysial and IgA anti-tissue glutamase antibodies associated with? IgA anti-endomysial and IgA anti-tissue glutamase are associated with celiac disease. What disorder is anti-basement membrane antibodies associated with? Anti-basement membrane antibodies are associated with Goodpasture syndrome. What disorder is anti-desmoglein antibodies associated with? Anti-desmoglein antibodies are associated with pemphigus vulgaris. What disorder is antimicrosomal and anti-thyroglobulin antibodies associated with? Antimicrosomal and anti-thyroglobulin antibodies are associated with Hashimoto's thyroiditis. What disorders are anti-Jo1 and anti-SRP and anti-Me2 antibodies associated with? These antibodies are associated with polymyositis and dermatomyositis. What disorder is the anti-SSA or anti-Rho antibodies associated with? Anti-SSA or anti-Rho antibodies are associated with Sjogren's syndrome. What disorder is the anti-SSB or anti-La antibodies associated with? Anti-SSB or anti-La antibodies are also associated with Sjogren's syndrome. What disorder is the anti-U1 ribonucleoprotein antibody associated with? Anti-U1 ribonucleoprotein antibody is associated with mixed connective tissue disease. What disorder is the anti-smooth muscle autoantibody associated with? Anti-smooth muscle autoantibody is associated with autoimmune hepatitis. What disorder is the anti-glutamate decarboxylase antibody associated with? Anti-glutamate decarboxylase antibody is associated with type 1 diabetes mellitus. What disorder is the C-ANCA antibody associated with? The C-ANCA antibody is associated with granulomatosis with polyangiitis, which is also known as Wegener's granulomatosis. What disorders is P-ANCA autoantibody associated with? P-ANCA autoantibody is associated with microscopic polyangiitis and Churg-Strauss syndrome. Infections and immunodeficiency. What bacterial complication may arise if you are T-cell deficient? With T-cell deficiency, you are more prone to sepsis. Name the common bacterial infections seen with B-cell deficiency. With B-cell deficiency, a patient would be more prone to encapsulated bacterial infection, and these include Streptococcus pneumoniae, Haemophilus influenza type B, Neisseria meningitidis, Salmonella, Klebsiella pneumoniae, and Group B strep. What are granulocytes? Granulocytes are white blood cells that have granules in their cytoplasm. There are three granulocytes. One is basophils, the second is eosinophils, and the third is neutrophil. And this can be remembered with the mnemonic BEN, B-E-N. Name the common bacterial infections seen with granulocyte deficiency. With granulocyte deficiency, you see infections with Staphylococcus, Burkholderia cepaceae, Serratia, and Nocardia. Name the common bacterial infections seen with complement deficiency. With complement deficiency, you are more prone to Neisseria infection, and this is because with complement deficiency, there is no formation of the membrane attack complex. Name the common viral infections seen in T-cell deficiency. With T-cell deficiency, you are more prone to viral infections such as cytomegalovirus, Epstein-Barr virus, varicella-zoster virus, and there can also be chronic infections with respiratory and GI viruses. Name the common viral infections seen with B-cell deficiency. B-cell deficiency can lead to viral infections such as enteroviral encephalitis and poliovirus. One thing to note here is that in these patients, live vaccines are contraindicated, and the reason for this is because that live attenuated vaccine may revert to a virulent or harmful form. Name the common fungal infection seen with T-cell deficiency. The common fungal infections seen with T-cell deficiency includes Candida and Pneumocystis pneumonia. What common parasitic infection is seen with B-cell deficiency? The parasitic infection that can occur with B-cell deficiency are GI infections due to giardias, and the reason for this is because there would be no IgA formation by the plasma cells. Name the fungal infections that can occur due to granulocyte deficiency. The fungal infections that can occur due to granulocyte deficiency is Candida and Aspergillus infections. So to note about the infections and immunodeficiency is that B-cell deficiencies tend to produce more recurrent bacterial infections, whereas T-cell deficiency will produce more fungal and viral infections. Immune deficiencies. Name all the B-cell disorders. B-cell disorders include X-linked Bruton's agammaglobulinemia, selective IgA deficiency, and common variable immunodeficiency. What is the genetic defect that leads to Bruton's agammaglobulinemia? In Bruton's agammaglobulinemia, there is a defect in the BTK gene, which is a tyrosine kinase gene, and this leads to no B-cell maturation. So in this condition, because the maturation is the problem, you still do have a normal amount of pro-B cells, but you don't have mature B cells. What type of inheritance is Bruton's agammaglobulinemia? Bruton's agammaglobulinemia is an X-linked recessive condition, so therefore it occurs mostly in boys. Describe the presentation of Bruton's agammaglobulinemia. Bruton's agammaglobulinemia will generally present with a boy that has recurrent bacterial infections after 6 months of age. And the reason for this is that after 6 months, if an infant is no longer breastfeeding, they will have a decrease in the maternal IgG, therefore they are now required to make their own, but because they have that maturation defect, they will no longer be able to produce mature B cells and therefore decrease also in the immunoglobulins. So basically that is what happens, because after 6 months of age, the infant is no longer able to opsonize the bacteria because of that deficiency of IgG. What are the findings observed in Bruton's agammaglobulinemia? In Bruton's agammaglobulinemia, you see normal pro-B cells as we already mentioned, but because the maturation is the defect, there is going to be a decrease in the mature B cells, and further downstream, that would also lead to a decrease in all immunoglobulin classes. What is selective IgA deficiency? Selective IgA deficiency is the most common primary immunodeficiency. The mechanism of this is unknown, but it's generally thought that the problem is B cells are not able to differentiate into plasma cells, therefore leading to decrease in production of antibodies. Describe the presentation seen with selective IgA deficiency. Patients with selective IgA deficiency are generally asymptomatic, but you may see cases of sinopulmonary infections, GI infections, and autoimmune disease. One important thing to remember about selective IgA deficiency is that if these people are given IgA-containing blood products, they may have a life-threatening anaphylactic reaction. What are the common findings observed in selective IgA deficiency? The findings of selective IgA deficiency includes low IgA, which is lower than 7 mg per deciliter, but the levels of IgG, IgM, and IgG vaccine titers are all going to be normal. With selective IgA deficiency, you could also have a false positive beta-HCG test due to the presence of heterophile antibodies, and that will cause the false positive pregnancy test. What is common variable immunodeficiency or CVID? Common variable immunodeficiency is a condition that has many causes, but ultimately the problem is with B cell maturation. And if B cells are not able to mature, they cannot make plasma cells, and therefore ultimately leading to also decrease in the immunoglobulins. Describe the presentation seen with common variable immunodeficiency. Common variable immunodeficiency is something that is acquired in the 20s and 30 age range, and it is thought that it's due to the mutation in genes that are involved in the development and function of B cells. These patients would be at an increased risk of autoimmune disease as well as sinopulmonary infections, and lymphoma is the most common cause of death in these patients. What findings are observed in common variable immunodeficiency or CVID? In common variable immunodeficiency, you see normal amount of B cells, but their maturation is defective, so there would be a decrease in the plasma cells and therefore decrease in immunoglobulins. Immune deficiencies, T-cell disorders. Name all the T-cell disorders. T-cell disorders includes DiGeorge syndrome or thymic aplasia, interleukin-12 receptor deficiency, hyperimmunoglobulin E syndrome or Job syndrome, and chronic mucocutaneous candidiasis. What defect leads to DiGeorge syndrome? DiGeorge syndrome happens when there is a deletion in chromosome 22q11, and this leads to the failure to develop third and fourth pharyngeal pouches, leading to that thymic aplasia. Describe the presentation seen with DiGeorge syndrome. In DiGeorge syndrome, the patient will develop tetany, and this is due to hypocalcemia. The reason this happens is because of that failure to develop third and fourth pharyngeal pouches. This would lead to failure to develop thymus and the parathyroid, which will lead to decrease in parathyroid hormone, as well as decrease in calcium, leading to hypocalcemia and therefore tetany. Other presentations of DiGeorge syndrome includes recurrent viral and fungal infection due to T-cell deficiency. And the reason for T-cell deficiency is, as we know, that T-cells mature in the thymus. So if you don't have a properly functioning thymus, it makes sense that there would be T-cell deficiency. Also, other presentation of that failure in third and fourth pharyngeal pouches includes cleft palate, congenital heart, and great vessel defects. What are all the findings observed in DiGeorge syndrome? The findings of DiGeorge syndrome includes thymus and parathyroid will fail to develop. This leads to decrease in T cells, decrease in parathyroid hormone, and decrease in calcium. You can also see this on chest x-ray where you would see an absent thymic shadow. Describe what problems occur with interleukin-12 receptor deficiency. As we mentioned earlier in the chapter, interleukin-12 induces the differentiation of T cells into T helper 1 cells. So therefore, deficiency of interleukin-12 receptor would lead to decrease in T helper 1 response. Describe the presentation of interleukin-12 receptor deficiency. With interleukin-12 receptor deficiency, you have decrease in T helper 1 response, and this leads to disseminated mycobacterial infections. What finding is observed with interleukin-12 receptor deficiency? We just said that interleukin-12 receptor deficiency will lead to a decrease in T helper 1 response. And as we mentioned earlier in the chapter, that T helper 1 cells are what secretes interferon gamma. So therefore, interleukin-12 receptor deficiency will lead to a decrease in interferon gamma. What is hyper-IgE syndrome or Job syndrome? Hyper-IgE syndrome or Job syndrome is when T helper 1 cells fail to produce interferon gamma, and this failure in production of interferon gamma leads to inability of neutrophils to respond to chemotactic stimuli. Describe the presentation of hyper-IgE syndrome. The presentation observed with a person with hyper-IgE syndrome or Job syndrome includes coarse feces, cold or non-inflamed staphylococcal abscesses, retained primary or baby teeth, increase in IgE, and dermatologic problems such as eczema. What is the mnemonic to help you remember Job syndrome or hyper-IgE syndrome? The mnemonic, just remember the word faded, and that's F-A-T-E-D. So F, which stands for coarse feces, A is for abscesses, T is for baby teeth, E is for increase in immunoglobulin E, and D is for dermatologic problems. Another way to also help you remember this problem is just remember that it's called Job syndrome after the biblical character Job, whose faithfulness in God was tested after he was given skin sores and pustules. What is chronic mucocutaneous candidiasis? Chronic mucocutaneous candidiasis is an immune disorder of the T cell, and as we mentioned earlier in this chapter that with T cell dysfunction you are more likely to get viral and fungal infections, so therefore with this condition there is recurrent Candida albicans infections of the skin and mucous membrane. Immune deficiencies, B and T cell disorders. Name all the B and T cell disorders. The B and T cell disorders include severe combined immunodeficiency or SCID, ataxia telangiectasia, hyper-IgM syndrome, and Wiskott-Aldrich syndrome. Name the two important types of severe combined immunodeficiency or SCID. The two important types include the X-linked severe combined immunodeficiency and the adenosine deaminase deficiency. What is X-linked severe combined immunodeficiency? X-linked severe combined immunodeficiency is the most common type of SCID. In this immunodeficiency disorder, the body produces very few T cells and natural killer cells. In this condition, there is also defective interleukin-2 receptor, and as we mentioned earlier in this chapter, that interleukin-2 is what stimulates the growth of helper, cytotoxic, and regulatory T cells. What is adenosine deaminase deficiency? Adenosine deaminase deficiency is the most common type of SCID. In this condition, there is defective adenosine deaminase enzyme, and that leads to inhibition of another enzyme which is called ribonucleotide reductase, and with the ribonucleotide reductase being inhibited, This leads to lymphocyte proliferation is inhibited and therefore immune system is compromised. Describe the presentation seen with severe combined immunodeficiency. When thinking about severe combined immunodeficiency disorder, just think about the movie Bubble Boy. In that movie, a person is inside a bubble just because they do not want to catch any sort of infection because they have this condition SCID. So the presentation of a person with SCID includes recurrent viral, bacterial, fungal, and protozoal infections, along with failure to thrive, chronic diarrhea, and thrush. In these patients, there is also going to be absence of the thymic shadow, germinal centers on lymph node biopsy, and B cells on peripheral blood smear. What is the treatment option for severe combined immunodeficiency? The treatment option is bone marrow transplant. What is ataxia telangiectasia? Ataxia telangiectasia is a rare neurodegenerative inherited disease causing severe disabilities. Ataxia refers to a poor coordination and telangiectasia to small dilated blood vessels, both of which are hallmarks of this disease. What defect causes ataxia telangiectasia? Ataxia telangiectasia occurs due to defect in the ATM gene, which codes for DNA repair enzyme. What kind of problems are caused by this defect in the ATM gene? Defect in this ATM gene leads to a triad of cerebellar defects which causes ataxia, spider angiomas, which is a type of telangiectasia, and IgA deficiency. What lab finding is commonly seen in ataxia telangiectasia? In approximately 95% of cases of ataxia telangiectasia, there is an increase in alpha-fetoprotein, but the reason that causes this increase is not yet known. What is the defect in hyper-IgM syndrome? In hyper-IgM syndrome, there is a defective CD40 ligand on helper T cells. When CD40 ligand is defective, you are not able to complete that second signal of B cell activation and class switching, therefore no class switch occurs. Describe the clinical presentation seen with hyper-IgM syndrome. In hyper-IgM syndrome, you often see patients with severe pyogenic infections early in life. What lab findings are seen with hyper-IgM syndrome? In hyper-IgM syndrome, as the name suggests, there is going to be an increase in the level of IgM, but there is going to be a decrease in immunoglobulin G, immunoglobulin A, and immunoglobulin E. Basically, what ends up happening in this condition is that B cells will keep making IgM antibodies, and the reason for this is because they can't class switch, and therefore they cannot make any other antibodies, so there's going to be an overproduction in IgM, but an underproduction in all the other antibodies. What is Wiskott-Aldrich syndrome? Wiskott-Aldrich syndrome is a rare X-linked recessive condition in which the WAS gene on the X chromosome is defective, and this leads to T cells that are unable to recognize the actin cytoskeleton. So basically, this condition is going to lead to immunodeficiency as well as reduced ability to form blood clots. Describe the presentation seen in Wiskott-Aldrich syndrome. Wiskott-Aldrich syndrome commonly presents in males because it's an X-linked recessive condition. You will also see a triad of thrombocytopenic purpura, infections, and eczema. And you can remember this by the mnemonic TIE, T-I-E, for thrombocytopenic purpura, infections, and eczema. What lab findings are observed in Wiskott-Aldrich syndrome? In Wiskott-Aldrich syndrome, you see an elevated levels of immunoglobulin E and IgA, but you see a decrease in the level of IgM. In this condition, because there is a decrease in the ability to form clots, you would also see thrombocytopenia, which is low platelet counts. Immune deficiencies, phagocyte dysfunction. Name all the phagocyte dysfunction disorders. The phagocyte dysfunction disorders include leukocyte adhesion deficiency type 1, Chidiac-Higashi syndrome, and chronic granulomatous disease. What is the defect in leukocyte adhesion deficiency type 1? In leukocyte adhesion deficiency type 1, there is a defect in the LFA1 or leukocyte function-associated antigen 1 integrin CD18 protein on phagocytes. So basically, this is a gene mutation that encodes CD18, and CD18 is what's normally found on the cell surface of LFA1. What are the signs and symptoms of leukocyte adhesion deficiency type 1? The signs and symptoms include recurrent bacterial infections, absent pus formation, and delayed separation of the umbilical cord. What lab findings are observed in leukocyte adhesion deficiency type 1? In this condition, because of the LFA1 deficiency, neutrophils are unable to adhere and to migrate out of the blood vessels, and therefore their blood count is going to be high, hence neutrophilia. What is Chidiac-Higashi syndrome? Chidiac-Higashi syndrome is an autosomal recessive condition. In this, there is a defect of the lysosomal trafficking regulator gene, or LYST, and this leads to microtubule dysfunction and phagosome-lysosome fusion, and therefore leading to decrease in phagocytosis. What are the signs and symptoms associated with Chidiac-Higashi syndrome? In Chidiac-Higashi syndrome, due to that decrease in phagocytosis, there is recurrent pyogenic infections by staphylococci and streptococci, along with partial albinism and peripheral neuropathy. What lab findings are seen in Chidiac-Higashi syndrome? In Chidiac-Higashi syndrome, you see giant granules or giant inclusion bodies in neutrophils. What is chronic granulomatous disease? Chronic granulomatous disease is an inherited disease in which there is a lack of NADPH oxidase, and this is an enzyme that is needed to produce reactive oxygen species such as superoxide, which is used to kill ingested pathogens. In this condition, there is also the absence of the respiratory burst in neutrophils. Describe respiratory burst. Respiratory burst is the rapid release of reactive oxygen species to kill off pathogens. What kind of infections are commonly seen in chronic granulomatous disease? With chronic granulomatous disease, there is increased susceptibility to catalase-positive organisms such as Staph aureus, E. coli, and Aspergillus. What are the two diagnostic tests for chronic granulomatous disease? The two tests used to diagnose chronic granulomatous disease includes dihydrorhodamine flow cytometry test, or DHR flow cytometry, along with nitroblue tetrazolium test. What are grafts or grafting? Grafts or grafting refers to a surgical procedure to move tissue from one side of the body to another or from one person to another without bringing its own blood supply with it. Name all the different types of grafts. The different type of grafts includes autograft, synergic graft, allograft, and xenograft. One thing to remember about grafts is that the higher the HLA compatibility, the higher chance of success of the graft, and graft from self-tissue has the highest HLA compatibility. What is autograft? Autograft is when the tissue that's used in the graft is from self. What is synergic graft? Synergic graft is the tissue is from an identical twin or a clone. What is allograft? Allograft is when the tissue that's used is from a non-identical individual of the same species. What is xenograft? Xenograft is when the tissue that's used is from a different species. Transplant rejection. What is transplant rejection? Transplant rejection is something that occurs when a transplanted tissue is rejected by the recipient's immune system, and in this process, the immune system will destroy the transplanted tissue. This effect can be lessened with immunosuppressant drugs after the transplant. What are the different types of transplant rejections? The different type of transplant rejection includes hyperacute, acute, chronic, and graft versus host disease. Describe the time frame of rejection seen with these different type of rejections. Hyperacute rejection occurs within minutes, acute rejection happens weeks later, chronic rejection happens months to years later, and graft versus host can have variable time frames. Describe the pathogenesis of hyperacute transplant rejection. Hyperacute transplant rejection is antibody-mediated. It's a type 2 hypersensitivity reaction, and this occurs due to the presence of performed anti-donor antibodies in the transplant recipient. What are the clinical features observed with hyperacute rejection? In hyperacute rejection, if the tissue is left implanted, it will occlude the graft vessels, and this will cause ischemia and necrosis in that area. Describe the pathogenesis of acute transplant rejection. Acute transplant rejection is cell-mediated due to cytotoxic T lymphocytes reacting against foreign M8Cs. This type of rejection can be reversible with immunosuppressants such as cyclosporin and muromonab CD3. What are the clinical features observed with acute transplant rejection? Acute transplant rejection may cause vasculitis of the graft vessels with dense interstitial lymphocytic infiltrate. Describe the pathogenesis of chronic transplant rejection. In chronic transplant rejection, class 1 M8C non-self is perceived by the cytotoxic T lymphocytes as class 1 M8C self, presenting a non-self antigen. So because of this mismatch, the cytotoxic T lymphocytes will attack the vessels and start causing damage. What are the clinical features observed with chronic transplant rejection? Chronic transplant rejection is unfortunately irreversible. There is T-cell and antibody-mediated vascular damage that occurs, which leads to obliterative vascular fibrosis. Chronic transplant rejection can also lead to fibrosis of graft tissue and blood vessels. Describe the pathogenesis of graft versus host disease. In graft versus host disease, the grafted immunocompetent T cells will proliferate and they would destroy and reject the host immunocompromised cells. So basically, the grafted T cells will proliferate and they would think that the host cells are foreign cells and therefore leading to destruction and eventually organ dysfunction. What clinical features are observed with graft versus host disease? In graft versus host disease, you would see maculopapular rash, jaundice, hepatosplenomegaly, and diarrhea. One additional note about graft versus host disease is that this normally occurs in bone marrow and liver transplant because these are the organs that are rich in lymphocytes. Immunology, immunosuppressants. What is the mechanism of action of cyclosporin? The mechanism of action of cyclosporin is to bind with cyclophilin, which is a cytosolic protein. This would form the cyclosporin-cyclophilin complex, and this complex blocks the differentiation and activation of T cells by inhibiting calcineurin. And that calcineurin in normal circumstance is what's responsible for activating the transcription of interleukin-2. So therefore, calcineurin inhibition would prevent the production of interleukin-2 and its receptors. What is the clinical use of cyclosporin? The clinical use of cyclosporin is to suppress organ rejection after transplantation, and it's also effective in certain autoimmune disorders. What are the side effects and toxicities of cyclosporin use? Cyclosporin use can lead to nephrotoxicity, hypertension, hyperlipidemia, hyperglycemia, tremors, gingival hyperplasia, and hirsutism. Define hirsutism. Hirsutism is just an abnormal growth of hair on a person's face or body. What is the mechanism of action of tacrolimus? The mechanism of action of tacrolimus is similar to cyclosporin. It binds to the FK binding protein and it will eventually inhibit calcineurin. And as we mentioned earlier, that inhibition of calcineurin will lead to decrease in production of interleukin-2. What is the clinical use of tacrolimus? Tacrolimus is a potent immunosuppressive agent used in organ transplant recipients to prevent organ rejection. What are the side effects and toxicities of tacrolimus? The side effects and toxicities of tacrolimus are similar to cyclosporin, where there is nephrotoxicity, hypertension, hyperlipidemia, hyperglycemia, and tremors. But the only thing that does not happen with tacrolimus is gingival hyperplasia and hirsutism. Those only happens with cyclosporin. So those are basically the only two differences between the side effects and toxicities of tacrolimus and cyclosporin. What is the mechanism of action of sirolimus or rapamycin? The mechanism of action of sirolimus or rapamycin is to inhibit T-cell proliferation in response to interleukin-2. And even though this name sounds similar to tacrolimus, it does not inhibit calcineurin, but instead this drug inhibits mTOR, which stands for mammalian target of rapamycin. What are the clinical uses of sirolimus? Sirolimus is used as immunosuppression after kidney transplantation in combination with cyclosporin and corticosteroids. It's also used with drug-eluting stents. What are the side effects and toxicities seen with sirolimus? The side effects and toxicities of sirolimus includes hyperlipidemia, thrombocytopenia, and leukopenia. What is the mechanism of action of azathioprine? Azathioprine is an anti-metabolite precursor of 6-mercaptopurin that interferes with the metabolism and synthesis of nucleic acid. So because this interferes with nucleic acid production, this becomes a toxic drug for proliferating lymphocytes, and this is why it works great as an immunosuppressant. What are the clinical uses of azathioprine? Azathioprine can be used after kidney transplantation. It can also be used for autoimmune disorders, including glomerulonephritis and hemolytic anemia. What are the side effects and toxicities associated with azathioprine? Azathioprine, because of its effect on nucleic acid, it can lead to bone marrow suppression. What drug-to-drug interactions should be avoided when taking azathioprine? Azathioprine's active metabolite is mercaptopurin, and mercaptopurin is metabolized by xanthine oxidase. So if you're taking drugs such as allopurinol to treat gout in which you would need to inhibit xanthine oxidase, that means that you would not be able to metabolize mercaptopurin. That means the levels of mercaptopurin will increase, and therefore toxicities of azathioprine would also be greater. So basically, when taking azathioprine, you shouldn't take any drugs that would inhibit xanthine oxidase because that would increase the level of mercaptopurin, leading to greater toxicities. Neuromonab CD3, also known as OKT3. What is the mechanism of action of neuromonab CD3 or OKT3? OKT3 is a monoclonal antibody that binds to the CD3 protein on the surface of T cells. It blocks the cellular interaction with CD3 protein and therefore leading to prevention of T cell signal transduction. What is the clinical use of OKT3? OKT3 is used as an immunosuppressant after kidney transplantation. What side effects and toxicities are observed with neuromonab CD3 or OKT3? The side effects and toxicities include cytokine release syndrome, and the reason this happens is when this monoclonal antibody first binds to CD3, it will cause T cells to release massive amount of cytokines, hence the name cytokine release syndrome. One additional thing to note about this drug is that there can also be hypersensitivity reaction that can occur. Recombinant cytokines and clinical uses. What is the clinical use of aldesleukin or the recombinant form of interleukin-2? Aldesleukin or interleukin-2 recombinant form is used in renal cell carcinoma and metastatic melanoma. What is the clinical use of epoetin alpha or erythropoietin? Epoetin alpha or erythropoietin is used to treat anemias, especially due to renal failure. What is the clinical use of the granulocyte colony stimulating factor filgrastim? Filgrastim is used to recover bone marrow function, which may be caused by suppression or in cases of some diseases. What is the clinical use of the granulocyte macrophage colony stimulating factor sargramostim? Sargramostim is used to also recover bone marrow function, which may be caused due to suppression or due to some disease conditions. What are the clinical uses of alpha interferon? Alpha interferon is used in hepatitis B and C infection, Kaposi sarcoma, leukemias, and with malignant melanoma. What is the clinical use of beta interferon? Beta interferon is used in multiple sclerosis. What is the clinical use of gamma interferon? Gamma interferon is used in chronic granulomatous disease. What is the clinical use of oprelvicin, which is a recombinant form of interleukin-11? The clinical use of oprelvicin is thrombocytopenia. What is the clinical use of thrombopoietin? As the name suggests, thrombopoietin is to treat thrombocytopenia. Therapeutic antibodies. What is the target site for neuromonab CD3 or OKT3? Neuromonab CD3 or OKT3 targets CD3 protein on the surface of T cells. What is the clinical use of neuromonab CD3 or OKT3? Neuromonab CD3 or OKT3 is used to prevent acute transplant rejection. What does digoxin immune Fab target? Digoxin immune Fab targets digoxin to neutralize it because it's an antidote for digoxin intoxication. What does infliximab target? Infliximab targets TNF-alpha because it's a monoclonal antibody that binds to TNF-alpha. What is the clinical use of infliximab? Infliximab is used in Crohn's disease, rheumatoid arthritis, psoriatic arthritis, and ankylosing spondylitis. What does adalimumab target? Adalimumab, just like infliximab, also targets TNF-alpha since it also is a monoclonal antibody that binds to TNF-alpha. What are all the clinical uses of adalimumab? Adalimumab is also used for Crohn's disease, rheumatoid arthritis, and psoriatic arthritis. What does abciximab target? Abciximab targets glycoprotein 2B and 3A and will prevent its expression, and in this way will prevent platelet-to-platelet binding. What are all the clinical uses of abciximab? Abciximab prevents cardiac ischemia and unstable angina and is useful in patients treated with percutaneous coronary intervention. What does trastuzumab or Herceptin target? Trastuzumab or Herceptin targets and inhibits HER2 receptors. What is the clinical use of trastuzumab or Herceptin? Trastuzumab or Herceptin is used to treat HER2 overexpressing breast cancer. What does rituximab target and inhibit? Rituximab targets and inhibits the CD20 surface protein. What is the clinical use of rituximab? Rituximab can be used to treat B-cell non-Hodgkin's lymphoma. What immunoglobulin does omalizumab target and inhibit? Omalizumab targets and inhibits immunoglobulin E. What is the clinical use of omalizumab? Omalizumab is used as an additional line of treatment for severe asthma.